Assalamu alaikum dear students. I hope you all are good, and today we are starting with the photorespiration. Yesterday we have read the Calvin cycle in which carbon dioxide is fixed to give glucose. Fine, but in photorespiration what happens? This RUBP, the primary carbon dioxide acceptor, it combines with oxygen in presence of Rubisco. Rubisco, you know, it is a bifunctional enzyme. It behaves as a carboxylase in presence of carbon dioxide, and in presence of oxygen it behaves as an oxygenase. Means it catalyzes the reaction of conversion of combination of oxygen plus ribulose bisphosphate and gives phosphoglycolate. This phosphoglycolate cannot be used in the chloroplast. So this photorespiration is also called a wasteful process because it produces the products which are not used in this chloroplast. As a result, it reduces the efficiency of the photosynthesis. So what has to be done? This phosphoglycolate, it gets transferred or it gets passed away to the peroxisome, which is the another cell organelle, and here this glycolate it enters here the glycolate cycle. Here you can see this cycle is called the glycolate cycle. Now this glycolate, this changes in presence of oxygen into glyoxylate, and these reactions take place in the peroxisomes. Now this glycolate, it changes into glycine. It's an amino acid, and then this amino acid goes to mitochondria, and here in mitochondria it changes into serine. It gets changed into serine, and then this serine is transported from mitochondria into the peroxisome. Fine, and then the serine here changes into glycerate and then phosphoglycerate. Phosphoglycerate you see here, and in this case ATPs are also used and NADPH are also used. So it is a wasteful process. So it reduces the efficiency of photosynthesis by approximately 25%. Fine. In this process, oxygen is used and carbon dioxide is produced, and it takes place in presence of Rubisco, but Rubisco here behaves as an oxygenase. Fine. So coming to the next, that is C4 cycle. But before starting the C4 cycle, you must be knowing what is Krebs anatomy. Krebs anatomy, you know, it's found in grasses. Grasses are monocots and in a few dicots, like in maize and few dicots it's also seen. So what happens in the leaves of these monocots, what happens, special type of the anatomy is seen in the cells, that's called the Krebs anatomy. You see here the bundle sheath cells. Fine, they have large chloroplasts. You see here bundle sheath cells in the dark black. They have large chloroplasts and are surrounded by mesophyll cells. Here you can see the mesophyll cells are in a ring form, or you can say it is a flower like anatomy, flower like a wreath is here, and wreath in German is called Kranz. That's why it is called as Krebs anatomy. So here these are arranged in a circular manner around the bundle sheath cells, and these mesophyll cells have a very small chloroplasts as compared to the bundle sheath cells. So this, I'll tell you what happens in the C4 cycle. In some plants, there is a mechanism of carbon dioxide fixation other than the C3 cycle. They undergo C4 cycle. Fine. So the plants which undergo C4 cycle for carbon dioxide fixation have Krebs anatomy in their leaves. So coming to the C4 cycle or Hatch-Slack cycle, it is called as Hatch-Slack cycle because it was discovered by a scientist whose name two scientists whose names were Hatch and Slack. That's why it's called as Hatch-Slack cycle. Why it's called as C4 cycle? Because the first stable product in this is oxaloacetic acid. It's a 4-carbon product. It's a 4-carbon product, oxaloacetate. Fine. So here what happens when the carbon dioxide enters the cytosol of the mesophyll cells, it combines with phosphoenol pyruvic acid. Here I have written PEP stands for phosphoenol pyruvic acid. It gives oxaloacetate. Fine. Now this oxaloacetate moves into the chloroplast. Here this is the chloroplast and this is the cytosol. Now this oxaloacetate moves into the chloroplast and gives malate. Fine, these malate is produced, and now this malate by means of diffusion goes into the, here let me tell you, these bundle sheath cells and these mesophyll cells, they are interconnected by plasmodesmata. You have read in the unit 1 plasmodesmata. What are those are the pores connecting the cells. Fine, so here it moves by diffusion through these plasmodesmata and enter the bundle sheath cells. Here the malate gets converted. Here the here it's carboxylation, means carbon dioxide is added here in the chloroplast, but when it enters the chloroplast of the bundle sheath cells, it undergoes decarboxylation, means carbon dioxide is released in this process. Carbon dioxide is removed from this reaction, and as a result pyruvate is produced. When malate is decarboxylized, pyruvate is produced. Now this carbon dioxide enters here the Calvin cycle, normal C3 cycle. Here in presence of rubisco, it combines with ribulose phosphate, and then normal Calvin cycle takes place, and triose phosphate is produced here, that is phosphoglyceric acid, or 3-phosphoglycerate you can say, is produced, which forms the glucose later on. Fine, so here the normal Calvin cycle takes place, and here what happens to this pyruvate? It moves then it moves here in this chloroplast of the mesophyll cells and gets converted to phosphoenol pyruvic acid. Here again it's converted to phosphoenol pyruvic acid. So the process continues. So in which plants it occurs? It occurs in those plants which are found in hot and dry environment. Hot and dry environments. Why it takes place in the plants? Because they have to escape the process of photorespiration. As I have already told you, photorespiration wastes approximately 25%. It reduces the efficiency of photosynthesis by 5%. So in order to avoid the photorespiration, those the plants in the hot and dry environments undergo this process. Fine, so they fix carbon dioxide by C4 pathway. Fine, and I have already told you it's called as C4 pathway because the first product formed here is oxaloacetic acid or oxaloacetate, and it's 4 carbon compound. Fine, these are the organic acids. Malate is then transferred to chloroplast of the bundle sheath cells. Here this step is very important. Decarboxylation takes place and the carbon dioxide is released into the normal Calvin cycle. I hope I am clear with photorespiration and C4 cycle or Hatch-Slack cycle. So if there is anything, please let me know in the comments. I'll be answering your questions. Thank you very much, and please go through the lectures also.